And now I get to talk about my favorite topic ever, my gorgeous, alluring worms, vampire worms. During grad school, I got to discover, describe, and name these three worms, and I'm so excited to talk about them with you right now. So you might ask, why are these super cute worms called vampire worms? Because they're vampires. They have this awesome eversible proboscis right here that actually normally sits inside their body cavity. And then when they're hungry, it comes shooting out. And it's lined with these little nib-like structures on the end of this proboscis that are called papillae. And these structures allow them to latch on to the plumes of these tube worms that I talked about earlier. And these plumes have lots of blood in them. They're saturated with blood. So the papillae, aided by the eversible proboscis, latches onto that plume and then begins to drink the blood of these awesome tube worms. I got to use a scanning electron microscope in grad school, which gives us a lot better, when I say a lot, I mean more detailed images than a regular light microscope, because electrons are smaller than photons. So that's how we're able to see much more detail. So here's the head. Here's the head of the worm. Here is their eversible proboscis. When they're not feeding, this whole structure right here slides back into the body cavity. And here's an up-close picture of the papillae, of those little nib-like structures that latch on to the plume of the tube worm. And this whole structure right here kind of works like a straw. It's able to drink the blood of the other type of tube worm, of the other type of worm. So, sample collection. Sample collection happens during deep sea cruises. Fun fact about deep sea cruises is that new species of marine organisms are being discovered during virtually every biological deep sea cruise. So every deep sea cruise that goes out into the ocean finds new species. That's how many new species are out there waiting to be discovered. So when we collect a sample, we cut the worm in half, and then we save the anterior, the head of the worm, for morphological analysis. So a lot of these discerning characteristics and traits that we can identify these species with are on the head in the first few segments. So that's why we keep the head for morphological analysis. And then the tail end, the posterior end, we use for DNA analysis. So for DNA, we preserve the tail end in ethanol and alcohol, which preserves DNA. And then we preserve the anterior, the head, for morphological analysis in a different preserving agent called formalin or formaldehyde, which preserves morphology. So, genetic analysis. Here I am in my old lab in grad school at Scripps Institution of Oceanography. So first we must extract the DNA from the tissue of the worm, and then you run a polymerase chain reaction, which exponentially increases the DNA strands. It's kind of like copy paste, copy paste, copy paste for all of these DNA strands. So then there's a lot of DNA within our test tubes. And then we test the PCR product to make sure that our efforts did in fact work using gel electrophoresis. So if DNA is present, then we see these little cute lights right here. And that means that DNA is in fact present in our test tubes. And we were successful in amplifying the DNA. And then we're able to send off our DNA to get sequenced. And then with our sequences, we're able to make a phylogenetic tree. So with our genetic sequences, we're able to compare and contrast with like species or related species. So here is a whole tree, a whole compilation of different organisms within the same group called Phylodosidae. And then this whole group is denoted by this blue color. And then my Galapagomystides genus within the greater group is denoted by this pink color right here. So I can see that my worms are related to each other. You can see that this branch right here, this is called a branch. This branches into a whole separate group of related organisms. This is a genus. Okay. This star right here means that this was the type species. Galapagomystides aristotis was the very first species discovered within the Galapagomystides genus. So all living organisms have something called binomial nomenclature, thanks to our good old friend Carl Linnaeus. Humans are Homo sapiens. Homo is the genus, sapien is the species. My worms are Galapagomystides and then the species. So Galapagomystides is the genus. And then I got to name my new species. And I'm gonna switch to the next slide. Oops, I guess maybe the slide after this, I'll introduce my new species. Okay, so I got to use light microscopes and also scanning electron microscopes, which I talked about earlier, shortened to the acronym SEM, scanning electron microscope. So with light microscopes, I'm able to see color because photons carry color. And then with a scanning electron microscope, I'm able to see a lot more detail. But since electrons don't carry color, there's no light in electrons, we get a black and white image. Okay, so for my 3 new species, welcome to the world, Galapagomystides bob-pearsoni, Galapagomystides cathiae, and Galapagomystides patricki. Galapagomystides bob-pearsoni. I named this particular species after my dad because my dad has always had this very prolific mustache my whole life. And this worm, this new species of deep sea worm, with its elongated anterior cirri protruding from the first few segments of this species, were reminiscent of my dad's very prolific mustache that you can see from these pictures. Galapagomystides bob-pearsoni was collected from Tonga at To Kam Vent at Lau Back Arc Basin, just under 2 miles down. Galapagomystides cathiae, I named after my mom. This new species was collected from Fiji at White Lady Vent, North Fiji Basin at 1,990 meters, which is just over 1 mile down. Galapagomystides patricki, I named after my now fiance, Patrick. And this new species was collected from Costa Rica at Parita Seep, just under 1 mile down. And earlier this year, I got an email from my grad school advisor, Dr. Greg Rouse, telling me that he has named a new species of deep sea worm after me. It is the most incredible compliment I have ever gotten. And welcome to the world, Ophiotrocha kylae. As a wrap-up, the deep sea is strange. It's bizarre. There are very cool adaptations, and it is unexplored. So go explore it.